So the first thing that you really need to know is how a neuron works. Now, there are about 83 billion neurons in the human brain. But what's even more interesting is the complexity in which these special type of cells communicate with each other. Now, the first thing you need to do is learn a little anatomy about the cell. Now, there's the cell body, where you have the nucleus and a lot of other organelles. You have dendrites, and the dendrites bring in information to the cell body. We'll talk about that. We have an axon, which sends signals away, electrical signals away from the cell body. There's the myelin, which is kind of this fatty tissue that surrounds the axon, kind of insulates it, but also speeds up the conduction of that information. And you have the nodes of Ranvier. These are little empty spaces along the axon where there's not myelin. And finally, we get to the terminal end, and these can branch out just like dendrites do. So the signal is sent both electrically and chemically, and we're going to look at how. So first thing you need to know really about the cell is its polarity. Polarity simply means that the inside of the cell has a charge that's different than the outside. It's polar. Like our Earth is polar. Our Earth has a positive and negative side. A magnet has a positive and negative side. And a cell, a neuron, has a positive and negative side. We call that a membrane potential or polarity. The inside of the cell is negative compared to the outside of the cell. And what keeps it negative on the inside is pumping out a lot of an ion that's called sodium. Sodium is positively charged. So if you take a positively charged ion and pull it out of a neuron, then the inside is more negative than the outside. We're going to look at that in more detail. Here's one way that they've determined this polarity. It's really a negative 70 millivolts. If you took a voltmeter and put one side inside of an axon and the other side outside, extracellular versus intracellular, you would see that difference. Oftentimes they use a giant squid axon because it's actually very large compared to human axons in their neurons. So there's the cell membrane right there. Now there's a lipid bilayer. The lipid bilayer is shown there in pink. And what's interesting about this membrane is it is only semi-permeable. It only allows some things in. So sodium can't come in, at least not through the bilayer there. And you can see outside the cell there's a lot of sodium and there's a lot of chlorine. And inside the cell there's a lot of potassium. Now potassium is positively charged. But what really gives the negativity to the inside of the cell are proteins, oftentimes marked as A negative. These are larger molecules and they're negatively charged. Now sodium wants to come in. If you can say an ion wants something, first there's this pressure of diffusion. It's just going where it's not. We see that with a diffusion pressure, things go where they're not. But there's also electrostatic pressure, and that really is the pull to the negative on the inside. Like the positive and negative side of a magnet attract each other, sodium, positively charged, is attracted to the inside or the negative, but it's kept out by this bilayer. Again, inside is negative 70 millivolts in comparison to the outside. That's the polarity. So let's go through some terms. A cell, a neuron at rest, is negative 70 millivolts inside compared to the outside. We call that a resting polarity or resting potential. But if we remove that polarity, if we make it less negative on the inside, we depolarize. And we can depolarize by bringing positive ions in. So if I bring positive inside, the negative 70 millivolts will go negative 69, negative 68, negative 65, or all the way down. Now that is called a depolarization. Sodium coming in depolarizes the neuron. But we can also hyperpolarize a cell. For example, if we took potassium out, more positively charged out will hyperpolarize. Or chlorine in, that's negatively charged, that would hyperpolarize. So we'd go from a negative 70 millivolts to negative 71, 72, and on. So now if we look at this membrane, we see embedded in the membrane are these channels. These are large protein channels. Sometimes they're called gates. They open and they close. So if they're closed, they don't let things in. If they open, they do. For example, let's take a sodium channel. If a sodium channel is closed, sodium can't come in. But if a sodium channel is open, sodium comes rushing in. It comes rushing in because of diffusion. It comes rushing in because of electrostatic pressure, pulling into the negative. This is going to be extremely important when we understand how neurons send signals. So here's my little diagram of a bilayer with a protein. Sodium can't come in. But if it opens up, sodium can come on in. Now that action will depolarize the membrane, remove the polarity a little bit. Now, there are a couple of ways, well, there are several ways, but the two ways I want to talk about here of how these channels open up. There are some that are voltage-dependent ion channels. That means that they open up because of the polarity around that channel. It's a voltage change that opens them. Sometimes they're called voltage-gated, and I'm going to talk about those first. But you should also know about chemical-dependent or chemical-gated ion channels. These are opened kind of like with a key to a lock. The lock is on the channel, and the key is a chemical. The chemical in this case is a neurotransmitter. We can think of neurotransmitters such as dopamine, serotonin, acetylcholine, GABA, glutamate, and they act either directly on that channel or they work on something else to open up that channel. So there's voltage-gated that are open with voltage and chemical-gated that are open with chemicals. Well, here is my diagram. It's kind of complex here, so we're going to take some time going through it. If you look outside the cell, there's lots of sodium. I have that in orange there. There's also some potassium and chlorine, but I'm focused on the sodium. There's a lot of negative charge on the inside, but that's mostly due to the proteins. So here is a membrane at rest, is a negative 70 millivolts when we compare the inside to the outside. Now suppose a channel opened up. There's a sodium channel opening up, and lots of sodium come rushing in. Now that middle gate right there, that middle channel, sodium channel, is now right around it, is a little bit different in its polarity because of all the positive ions that come in. And maybe right around there, it's now not negative 70, but it's been depolarized a little bit, and now it's negative 65. Now that might be the voltage that this voltage-gated ion channel is sensitive to. When that sodium channel, when it reaches negative 70 millivolts around it, it also opens up. Voltage-gated opens because of voltage. Sodium comes rushing in. Another voltage-gated ion channel opens next to it, down the axon. Sodium comes in, and maybe behind it, the sodium channels close. But you can see the progression of a signal from the left to the right. I mean, that's what we're trying to do here. Let's look at it again. Ion channel opens up. Sodium comes in, causes this ion channel to depolarize. Sodium comes rushing in, causes the next ion channel to open. Sodium comes rushing in. We want to be able to send a signal from one neuron to another, and this is the way we do it. We send this electrical changes moving in one direction down an axon. Let's look at it again. Now in this one, I'm showing the cell body over here, and here's where the axon connects to the cell body. We call that the axon hillock. And you can see the sodium channels in there. These are voltage gated. I also have some myelin there, the myelin and the empty spaces, the nodes of Ranvier. And what happens is the signal of depolarization bounces down. And now we have a signal going from one end of the axon to the other, from the cell body down to the terminal. Let's watch that again. This is a depolarization going down. One depolarization wave affects the other, and we have a moving or a propagation of the action potential. Now, this kind of slide you can see in just about any book. You can see it online, and it's very important. I really want you to understand this. This graph, on the x-axis or the horizontal axis, we have time, very fast time. And on the vertical axis or y-axis, we have the polarity inside the cell. the potential difference between the inside and the outside. If we have a place where sodium channels open, they reach their threshold, because at rest it's -70, then they reach their threshold, a lot of sodium channels will all open and we'll have a mass depolarization right there. Look at all that sodium rushing in, right? Keep that in mind because we're going to then have to repolarize. So what we need to do is we need to repolarize the cell. Sodium depolarizes the membrane and potassium repolarizes it. So sodium channels open up, sodium comes rushing in. This is a depolarization. But then potassium channels open very quickly after that because their voltage is met and potassium leaves. If potassium leaves, we are repolarizing, especially if sodium channels close. Sodium's channels close, we can't depolarize anymore. Potassium channels open, we repolarize. So let's look at this. Sodium has rushed in and we're depolarizing. Lots of sodium rushing in. But you can see here that potassium channels begin to open and begin to repolarize. Sodium channels close, so we can't depolarize anymore. But potassium channels remain open, leaving. Potassium leaves, therefore repolarizing the cell. And we get back to rest. We even overshoot a little bit. So let's look at the signal and how it really works. In green, we have the sodium channels. In red, we have the potassium channels. And what we get is a depolarization followed by a repolarization. Depolarization, repolarization. Depolarization, repolarization. The depolarization wave, or kind of like an explosion, affects down the axon a little bit, but then we repolarize. Let's watch that again. Depolarization, repolarization, depolarization, repolarization, all the way down. So let's look at the neuron as a whole. The neuron is getting a lot of information from the dendrites. And if the area right at the axon hillock, if those sodium channels meet their threshold, then we begin the action potential. And the action potential bounces down the nodes of Ranvier, all the way down until we get to the terminal end. We call that an action potential. And in this case, it's bouncing from node to node, and we actually call that a saltatory conduction. Saltatory means to jump. Here's just another way of looking at it. Sodium comes in, depolarization. Potassium leaves, repolarization. So depolarization is when sodium channels open up and sodium comes rushing in. Sodium depolarizes the cell because it brings positive charges inside. Repolarization is caused by potassium channels open and potassium leaving. Depolarization followed by repolarization. However, we have a problem now. We have a lot of sodium in and a lot of potassium out. We have a lot of sodium in and a lot of potassium out. So we need to use some energy to pull sodium out of the cell and to pull potassium back in. Pull 3 sodiums out, pull potassium in. We get that energy by ATP. Now, I don't know if you remember from biology, but ATP is created from ADP during cellular respiration, breaking down of sugars. And this whole process of taking sodium out, bringing potassium in, takes energy, and that is in the conversion of ATP to ADP. Okay, let's watch it again. Sodium is being pumped out using ATP, using energy. This is why your brain needs oxygen and sugar. This is a very expensive process in terms of energy. So now we're ready to go again. But keep in mind that the action potential is not caused by this. The action potential is caused by depolarization, sodium coming in, and that wave going down. So what happens when it gets at the end? What happens when that action potential finally reaches the terminal end of a axon? We get to an axon terminal, sometimes called an axon, a terminal button. Okay, so here we are at the end. We have the terminal end of an axon, on the left there, and a postsynaptic cell with some dendrites. The middle part is called the synapse. It's a space. There's actually a space between one neuron communicating to another. They don't touch each other. Not in this example anyway. So you have a presynaptic cell and a postsynaptic cell. Now waiting at the end of that presynaptic cell are vesicles. Vesicles are kind of like balloons. And they are holding neurotransmitters. And we're going to talk a lot more about them as the class progresses. But you can see there, those are the neurotransmitters such as glutamate or GABA or serotonin. Now, when the action potential comes down, depolarization, repolarization, it gets to some new ion channels, voltage-gated ion channels. These voltage-gated ion channels are calcium. They allow calcium to come in. And calcium plays a big role in causing the vesicles with the neurotransmitters to fuse with the presynaptic cell and spit out their neurotransmitters. This is called excitatory. Excitatory. So a neurotransmitter action potential comes down, and a calcium comes in, neurotransmitters are released, and they affect the postsynaptic cell. One more time. Action potential comes down, voltage-gated calcium channels open, calcium helps in exocytosis, in the release of neurotransmitters. This is the chemical process in the electrical chemical signal. So let's look at this in a big picture. The action potential goes down the axon and causes the release of neurotransmitters.
Boom, 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 gotta get that. 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 Boom, 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 that boom.
I'm always reminded of that boom, boom, boom going down the axon. Okay, so let's talk about the neurotransmitters. Neurotransmitters are, we'll go into a little bit greater detail when we talk about them in relationship to behavior. Dopamine, for example, plays a big role in addiction and motivation, and serotonin is thought to have a big role in moods. These are just chemicals that you're creating through enzymatic processes. You're going to package them in the vesicles, and you're going to take them down to the end of the terminal end of the axon, and wait there for the electrical signal to tell them to be released. So neurotransmitters fit a receptor like a specific key to a lock. Fits a receptor. Okay, so here is a kind of an added picture. We have the receptors, they are in green, and the chemicals, the neurotransmitters fit them like keys to a lock. And you can have the receptors on the cell body. You can have receptors on dendrites. You can even have receptors on axons. So here we go again. Action potential comes down, calcium comes in, causes the release of neurotransmitters, and then they go and fit in with these chemical gated ion channels. They aren't directed, they aren't pulled, they kind of fit because there are so many of them. Neurotransmitter comes in, fits, opens, and sodium comes in. Very simple. One last thing you should know, because this is going to be important when we talk about, for example, depression, is that the action potential can come down, calcium comes in, neurotransmitters released to the postsynaptic cell, but they can also come back in. They can also go through on the presynaptic cell, and those are sort of channels that pull neurotransmitters back in. Those are called reuptake channels, and you may have heard in the course of other courses something called selective serotonin reuptake inhibitors. These keep neurotransmitters like serotonin from being pulled back into the cell. So neurotransmitters can be released, but they also can be pulled back in. Now, my drawings are rather simple. I just have one axon going to one terminal, but the actual network of neurons in your brain are extremely complex. Neurons communicate to thousands of other neurons, and one neuron can have thousands of other cells affecting it at the dendrite level, at the cell level. These are extremely complex webs. Another very basic idea to understand is a neurotransmitter can have one of two effects on the other cell, on the postsynaptic cell. It can tell that cell to be excited. In other words, to increase the likelihood of action potentials. We call that a postsynaptic excitatory potential, EPSP. Glutamate, dopamine, serotonin, epinephrine, these are all excitatory neurotransmitters, though dopamine sometimes can be inhibitory. A neurotransmitter can go to a postsynaptic cell and tell that cell, don't fire so much, fire less often. Excitation and inhibition, and that's how your whole nervous system works based on excitation and inhibition. And we're going to look at a lot of these different neurotransmitters, such as GABA, serotonin, dopamine, and how they work. Okay, let's look at a few of them, some of the most common. Acetylcholine is extremely common, and it works on the neuromuscular junction is one place it works, but it's also throughout the brain. It can either be excitatory or inhibitory. Now, when we talk about the receptors, we give the receptors names. Acetylcholine's receptors are nicotinic and muscarinic. Norepinephrine is a neurotransmitter, and it is also in the brain, in the central nervous system. When we talk about their receptors as alpha and beta, and norepinephrine is an excitatory neurotransmitter. It causes the postsynaptic cell to fire more often. Dopamine, very, very common neurotransmitter. We'll talk about that when we talk about schizophrenia, attention deficit hyperactivity disorder, Parkinson's disease, addiction, and it can be both excitatory and inhibitory. Now, what's nice about these receptors is these are just D1, D2, D3, D4. And these different type of receptors will be important when we talk about antipsychotics. These are things that reduce psychosis, such as schizophrenia. Glutamate is the most common excitatory neurotransmitter, very important in learning. We'll talk about how cells bond together in a learning process. We have NMDA and AMPA receptors. You're going to eventually need to know these receptors as we talk about other material. Serotonin, 5-hydroxytryptamine, and these are 5-HT1, 5-HT2. Those are the types of receptors, both excitatory and inhibitory. GABA is the most common inhibitory neurotransmitter, and this will be very important when we talk about anxiety. And of course, the opioids, and those are given the Greek letters mu and lambda when they talk about the receptors. These are also inhibitory. Okay, when we talk about psychopharmacology, we often talk about antagonist and agonist. An antagonist is a drug that stops a neurotransmitter from doing its job. It blocks a receptor, it destroys a neurotransmitter, things like this. Oftentimes, people with schizophrenia, there's too much dopamine in certain areas, so they take a dopamine antagonist. Then there's the agonist, a drug that facilitates or helps a neurotransmitter to do its job. So cocaine is a dopamine agonist because it helps for more dopamine to be in the receptor, in the synapse, and get to receptors.